The world of nature is full of surprises. Some combinations are deadly, like mixing bleach and ammonia. Others, however, are surprisingly beneficial. One such curious pairing is snake venom and honey. Snake venom, a potent cocktail of toxins, is feared for its ability to cause paralysis and even death. Honey, on the other hand, is a golden liquid revered for its sweetness and medicinal properties. What happens when these two contrasting substances collide? This essay delves into the intriguing world of snake venom and honey, exploring their individual properties and the historical beliefs surrounding their unlikely partnership. Will uncover the scientific reality behind the folklore and examine whether this unusual pairing results in a toxic concoction or a potential source of healing. Join me as we separate fact from fiction and unravel the mysteries surrounding this fascinating subject. For centuries, humans have been fascinated by the power of natural remedies. Our ancestors experimented with various plants, animal products, and minerals to treat ailments and improve their well-being. Snake venom and honey, two readily available substances in many parts of the world, found their way into numerous folk remedies. These remedies, passed down through generations, often attributed magical or mystical properties to this unusual combination. In some cultures, snake venom mixed with honey was believed to possess potent healing properties, capable of curing anything from snake bites to arthritis. Others used it as an aphrodisiac or a way to enhance longevity. These beliefs, often rooted in observation and experimentation, highlight the human desire to harness the power of nature for therapeutic purposes. However, it's essential to approach such claims with a healthy dose of skepticism and examine them through the lens of modern science. Snake venom, a complex mixture of enzymes, proteins, and other molecules, is far more intricate than a simple poison. It's a sophisticated evolutionary tool, meticulously honed over millennia to subdue prey and aid in digestion. Each snake species boasts a unique venom cocktail, tailored to its specific diet and hunting strategies. Understanding the composition of snake venom is crucial to comprehending its effects, both detrimental and potentially beneficial. The potency and specific effects of snake venom vary drastically between species. Some venoms attack the nervous system, leading to paralysis, while others target the circulatory system, disrupting blood clotting and causing internal bleeding. The composition of snake venom determines its mode of action and the severity of its effects on the victim. This diversity in venom composition has fascinated scientists for decades, leading to ongoing research into its potential medical applications. Enzymes, the workhorses of biological systems, constitute a significant portion of snake venom. These proteins act as catalysts, accelerating chemical reactions within the body. In the context of venom, enzymes play a crucial role in breaking down tissues, aiding digestion, and facilitating the spread of toxins. One such enzyme, phospholipase A2, disrupts cell membranes, causing localized tissue damage and pain. Another key enzyme found in many snake venoms is hyaluronidase. This enzyme breaks down hyaluronic acid, a substance that helps hold cells together. By degrading hyaluronic acid, hyaluronidase increases tissue permeability, allowing other venom components to spread more rapidly throughout the victim's body. These enzymes, while potentially harmful in large quantities, have piqued the interest of medical researchers for their potential applications in targeted drug delivery and tissue regeneration. Toxins, the most well-known components of snake venom, are responsible for the debilitating and often lethal effects associated with snake bites. These molecules, typically proteins or peptides, interact with specific targets in the victim's body, disrupting vital physiological processes. Neurotoxins, as their name suggests, target the nervous system, interfering with nerve impulse transmission. This can lead to paralysis, respiratory failure, and even death. Cytotoxins, another class of toxins, directly attack and destroy cells. These toxins can cause severe tissue damage, necrosis, and organ failure. Hemotoxins, on the other hand, disrupt blood clotting mechanisms, leading to internal bleeding and hemorrhaging. The specific types and quantities of toxins present in snake venom determine its overall toxicity and the clinical manifestations of envenomation. Honey, a viscous golden liquid produced by honeybees from the nectar of flowering plants, has been a prized substance for millennia. Its sweetness has tantalized taste buds, while its remarkable properties have earned it a place of honor in traditional medicine across cultures. From ancient civilizations to modern times, honey has been revered as a natural healer, a symbol of purity and a source of nourishment. The journey of honey begins with industrious honeybees collecting nectar from blossoms. They store this sugary liquid in their honey sacs, where enzymes begin breaking down complex sugars into simpler ones. Back at the hive, the bees deposit the nectar into honeycombs, where further enzymatic action and evaporation concentrate the nectar into honey. The bees then seal the honeycombs with beeswax, preserving this liquid gold for future consumption. Honey's complex composition contributes to its unique flavor profile and remarkable properties. While the exact composition varies depending on the floral source, honey generally consists primarily of sugars, with fructose and glucose being the most abundant. These simple sugars provide honey with its characteristic sweetness and contribute to its energy-boosting properties. Beyond sugars, honey contains a variety of other components, albeit in smaller quantities, which contribute to its complexity. Enzymes derived from both the bees and the nectar source play a role in honey's digestion and contribute to its antioxidant activity. Honey also contains trace amounts of vitamins, minerals, amino acids, and antioxidants, which contribute to its nutritional value and potential health benefits. Beyond its delectable sweetness, honey's allure extends far and wide. For centuries, it has been recognized for its remarkable antibacterial properties. Its high sugar content creates an osmotic environment that inhibits the growth of many bacteria. Additionally, honey contains an enzyme called glucose oxidase, which produces hydrogen peroxide, a potent antimicrobial agent. Honey's viscosity also contributes to its wound-healing properties. Its sticky nature helps to create a protective barrier over wounds, preventing infection and promoting healing. Honey's antioxidant content further aids in wound repair by reducing inflammation and oxidative stress. These properties, combined with its soothing and moisturizing effects, have made honey a popular ingredient in traditional wound care practices. Honey's potential benefits extend beyond wound healing and antibacterial action. Emerging research suggests that honey may also play a role in modulating the immune system. Studies have shown that honey can stimulate the production of immune cells, such as macrophages and lymphocytes, which play crucial roles in fighting infections and diseases. Furthermore, honey's antioxidant properties may help to protect immune cells from damage caused by free radicals. Free radicals, those unstable molecules produced during normal metabolic processes, can damage cells and contribute to inflammation. Antioxidants, like those found in honey, neutralize free radicals, protecting cells from their harmful effects. The question of whether honey can neutralize snake venom is a tricky one, often clouded by folklore and anecdotal evidence. While honey possesses remarkable antibacterial properties, its ability to counteract the complex toxins found in snake venom is a different matter altogether. To understand why, we need to delve into the chemical nature of both substances. Snake venom, as we've established, is a potent cocktail of enzymes and toxins designed to disrupt physiological processes. Honey, on the other hand, is primarily composed of sugars, along with trace amounts of enzymes, vitamins, and antioxidants. The key question is whether any of these components can effectively neutralize the harmful effects of venom. The most common misconception is that honey's high sugar content can somehow denature venom, rendering it harmless. Denaturation refers to the disruption of a protein's structure, which can lead to a loss of function. While heat, acids, and certain chemicals can denature proteins, honey's sugar content is unlikely to have a significant denaturing effect on venom toxins. Venom toxins are often highly stable molecules, resistant to changes in pH or sugar concentration. Honey's sweetness, while delightful to our taste buds, is unlikely to disrupt the intricate three-dimensional structures of these toxins, leaving their function largely intact. Therefore, simply applying honey to a snakebite wound is unlikely to neutralize the venom's harmful effects. The belief in honey's ability to neutralize snake venom likely stems from its well-documented wound-healing properties. Honey has been used for centuries to treat wounds and prevent infections, thanks to its antibacterial and soothing properties. It's plausible that in some cases, applying honey to a snakebite wound may have helped to prevent secondary infections, leading to the misconception that it neutralized the venom itself. Furthermore, honey's viscosity may have played a role in slowing down the spread of venom in some cases. The thick, sticky nature of honey could potentially hinder the diffusion of venom through tissues, giving the body more time to mount an immune response. However, this effect is likely to be minimal and should not be mistaken for venom neutralization. While honey is unlikely to neutralize snake venom directly, emerging research suggests that it may hold some potential in mitigating the effects of envenomation. Scientists are exploring the possibility of using honey in combination with conventional anti-venom treatments to enhance their efficacy. One area of interest is honey's potential to reduce inflammation and tissue damage caused by snake venom. Certain compounds found in honey have shown anti-inflammatory properties in laboratory studies. Researchers are investigating whether these compounds could help to lessen the severity of venom-induced tissue damage, potentially improving outcomes for snakebite victims. The entwined history of snake venom and honey in traditional medicine stretches back millennia, weaving a fascinating narrative of human interaction with the natural world. Ancient civilizations, observing the potent effects of both substances, incorporated them into their healing practices, often attributing mystical or spiritual significance to their use. In ancient Egypt, where snakes held a prominent place in mythology and medicine, snake venom was revered for its perceived healing properties. The Egyptians believed that the venom of cobras, vipers, and other venomous snakes, when carefully administered, could cure a range of ailments from headaches to epilepsy. Honey, with its renowned antibacterial and wound-healing properties, was often combined with snake venom to create poultices, ointments, and elixirs. These remedies, often shrouded in ritual and secrecy, were passed down through generations of healers, forming an integral part of ancient Egyptian medical knowledge. The Ebers Papyrus, an ancient Egyptian medical text dating back to the 16th century BC, mentions several remedies containing both snake venom and honey, highlighting their significance in traditional Egyptian medicine. The use of snake venom and honey in traditional medicine extends far beyond ancient Egypt, appearing in various forms across cultures and continents. In traditional Chinese medicine, snake venom, known as shidan, has been used for centuries to treat a variety of conditions, including arthritis, rheumatism, and skin diseases. Honey, revered for its nourishing and harmonizing properties, was often incorporated into these remedies to enhance their efficacy and balance their potency. In Ayurvedic medicine, the ancient Indian system of healing, snake venom, known as Visha, is considered a powerful substance with both toxic and therapeutic properties. Ayurvedic practitioners believe that snake venom, when carefully processed and administered in minute doses, can stimulate the body's natural healing mechanisms and address a range of ailments from chronic pain to neurological disorders. Honey, a revered Ayurvedic remedy for its antibacterial, anti-inflammatory, and wound-healing properties, is often combined with snake venom to mitigate its toxicity and enhance its therapeutic effects. These examples illustrate the diverse ways in which cultures worldwide have incorporated snake venom and honey into their traditional medical practices. Modern science delves into snake venom and honey, separating fact from folklore. Evaluating traditional beliefs through scientific evidence is essential. Researchers explore their mechanisms and therapeutic applications. Snake venom holds potential medical applications. Venom peptides may become painkillers and cancer treatments. Honey's antibacterial and wound-healing properties are under study. The interaction between venom and honey remains largely unanswered. Preliminary studies suggest potential synergies, but more research is needed. The potential lies in harnessing their strengths while mitigating drawbacks. Scientific exploration often leads to unexpected discoveries. Traditional beliefs remind us of humanity's quest to harness nature's power. Delving deeper may uncover hidden synergies and therapeutic potentials. The future of medicine might lie in these unexpected pairings. Our exploration of snake venom and honey reveals nature's paradoxes. Snake venom, a toxic cocktail, contrasts honey, a healing elixir. Traditional medicine embraces their use. Modern science seeks understanding. Venom's complex enzymes target vital processes. Honey's antibacterial and wound-healing properties are explored. Its role in immune modulation is studied. Honey can't neutralize venom but shows potential in context. This intersection defies simplistic categorization. Dangerous contradictions may hold untapped potential. Unraveling these mysteries could lead to breakthroughs. The journey of exploration reveals wonders beyond the surface. Future medicine may lie in these unconventional pairings.